Today I will discuss about the assignment, the group assignment. Yeah. So regarding how to discover a novel gene using bioinformatics tool. Right. Before the era of bioinformatics, the scientists study protein using biochemistry and molecular biology. Right. The interested protein is purified from the culture medium based on enzymatic activity. And the purified protein is then sequenced in order to get the amino acid sequence. This is how biochemists study the protein and get the sequence of the protein. And then molecular biologists construct genomic DNA library and screen for positive clone based on enzymatic activity. This is how the molecular biologists clone the gene using genomic DNA library or PCR cloning. And then bioinformatics identify the novel gene using blast searching tool. You learned before in chapter 4, there are various of the blast tool that you can use in this assignment. This assignment will cover a lot of techniques from the blast to multiple sequence alignment to phylogenetic tree. Dr. Faria will cover next week regarding the phylogenetic tree. So, okay, for this assignment, you need to apply various techniques you have learned in bioinformatics courses. This is the key step to identify a novel gene. You can start with a protein sequence from any organism. Okay, for example, for this example, you can start with protein sequence. For example, if you like to start with beta globin from humans, and then you just need to get the amino acid sequence from NCBI database. Right? You can start with any protein sequence from any organism that you like. Okay? And then perform T blast N. T blast N is a type of blast, protein blast against translated DNA sequence from the database. And then perform T blast N against fully characterized database such as HPGS, EST database, or genome sequence database from a specific organism. Perform the search for certain genome from certain organism, selected organism, right? And then please take note, do not search against model organisms, for example, E. coli, yeast, Pichia, Pasteuris. This type of the organism has the genome sequence has been sequenced for quite some time, and then every single gene inside the genomic DNA has been annotated, right? And then after you perform the blast and then inspect the output, it should not match with 100% amino acid identity to protein in the database from the same species, right? And then this can be verified using BLAST-X, which is translated DNA blast against protein database, or using BLAST-P, protein blast against protein database. And at the end, you have to verify whether it is novel gene or not by performing BLAST-X or BLAST-P. So for this assignment, you have to employ both techniques to verify the novel gene. This is a various thing that Dr. Fiza has covered during her lecture, right? BLAST-P, BLAST-N, BLAST-X, T-BLAST-N, and T-BLAST-X. And then for this assignment, you need to use the technique of BLAST-P, BLAST-X, and T-BLAST-N. Okay? Okay, this is step 1. First, you have to choose your favorite protein. For example, in this example here, beta globin from human. Right? If you don't have the sequence, you can retrieve any amino acid sequence from NCBI database. This is just the example. You no need to follow this. You can start with your own target protein, your own interested proteins. In case you don't have the amino acid sequence, just go to NCBI database to retrieve the sequence based on the based on the knowledge you have learned in chapter 2, right? Access information from database, right? Okay, and then this is an example of the T plus N output. After you perform T plus N, and then you get something, and this output, right? And then from this output, you will see some of the hit producing significant e-value. Significant e-value, meaning that the e-value less than 0.05 for the case of blood P, right? 0.05, 0.05. And then you see 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, the first 9 give you a very significant e-value, close to 0. Right? And then the next, starting from sequence number 10 until 20 plus, you see the e-value is not significant. It starts from 0.2, which is slightly above the threshold e-value of 0.05. So in order to find a novel gene, I don't encourage you to choose the sequence from those producing significant e-value. Most probably will not get a novel gene based on if you choose any of the sequence from those sequences from are producing significant e-value. So I suggest you to choose the sequence that's slightly above the threshold value of 0.05. For example, here is 0.2. So this is 0.2, and then match to Aqualegia cDNA library. Right. In some cases, you may try more than one, and then just look at this example first. Okay. And after you click on the match, this is just now the highlight with yellow, and then this is the pairwise sequence alignments between human beta globin and Aqualegia globin. And then for BLAST-P, you can take only the subject sequence as input for BLAST-P. The next step is for you to verify whether this is a novel gene. It can be you can be taking the amino acid sequence from here by deleting the query sequence here. Just take a line for subject line, for this line, subject line, and then combine all the subject line, and then you will get the full sequence for your novel protein. In order to verify, and then you just take the subject line here, you combine the amino acid sequence from the subject lines here, and then you perform BLAST-P search. And then this is the first type of the verification technique. The second verification technique is using BLAST-X. So in order to get the DNA sequence, you can click on the accessor number, and then this will link you to gene bank record, and then you can get the accessor number for from the nucleotide sequence, right? Okay. After you remove the query line, and then take only the subject line, and then this amino acid for you to perform the search using BLAST-P, right? And then you may also click on the accessor number to retrieve the DNA sequence for BLAST-X. So both techniques you have to use for this assignment. Okay. And then after you perform the BLAST-P, and then the best two match are two proteins from Arabidopsis from Brassica. And since these two matches are not from Aqualegia, and we can conclude that no one has pre-identified our Aqualegia DNA as encoding a globin. So it is a novel, and we can identify, we can further study it further. Okay, the first two match to symbiotic, what not so clear what globin from Arabidopsis, and also match to non-symbiotic hemoglobin from Brassica. So the first two match is not the same as the sequence that you took, not the same as this Aqualegia. So we can conclude that this could be the novel, this could be the novel gene. So this is the first technique that you verify using BLASTP search by using just the amino acid sequence from the subject line. And then the second verification technique is using BLASTX. Okay, this one is from a nucleotide sequence from BLASTX. And then this is a result for BLASTP search. Same thing. The best match is not to Aqualegia proteins. And this sequence is indeed encodes for a novel protein. Right? The first match is to non-symbiotic hemoglobin, right? From Arabidopsis. Okay. Okay. The gene is not novel if it matches 100% to the same species. Okay, for example, just now you take the sequence, you start with this sequence, and you choose the sequence from Aqualegia cDNA library, and then after you perform BLASTX search, and then also the BLASTP search, and then if you get back the same thing from Aqualegia, and then with 100% match, meaning that this is not a novel protein. Right? And then the gene is novel if it matches less than 100% or even 100% but to a different species. So if after you perform BLASTX and also BLASTP, and then the first match is not belongs to Aqualegia, and then we can conclude that this is novel protein. Right? It can be 100% or less than 100%, but has to be different species. If no database match, meaning that the DNA or protein sequence is not homologous to the original query protein. Right? Okay. Understand now how to find a novel gene? How to do it?
Yes, doctor.
Yes, doctor.
Okay. Okay, it's very easy. And then we'll be in group. And then by today, I will upload the assignment to Ultrablast. And then how many of you in this class? 30, 49, 39, 50, 39, 49. How many of you in this class? 49, right?
$37, doctor. 37.
57? Yeah. 57, 57. Oh, a few of you missing. 57. 50?
37.
57. Okay. I divide you into how many groups? Do you prefer 6, 10? 10 group, okay. Later, I will post a grouping list to the Putra block. Divide into 10 groups. And then this is group assignment. Okay, in case you're not sure whether it's novel gene or not, and then you can text me, so I can double check it for you. Okay.
Okay, doctor.
Okay, doctor.
Okay, and then this is regarding the assignment. The practical side multiple sequence alignment. Okay, I don't have the bioedit with me because I'm using MacBook. There is no bioedit for the MacBook, but it is okay. The purpose is to perform cluster W. That is, and then I use the similar platform cluster W from EBI, and then I start with GDSL asterisk. This is the amino sequence for GDSL asterisk. Okay, and then before I perform the blast, I will detect the presence of the signal peptide. This is because the signal peptide with no function. The only function for signal peptide is to secrete the protein out from the cell into the culture medium. Once the protein is secreted out into the culture medium, and then the signal peptide will cleave away by signal peptidase, and then release the mature protein into the culture medium. And so, in order to check whether this GDSL assay consists of signal peptide or not, and then you can use signal P prediction to predict the presence of the signal peptide. So from the signal P software, and then it indicate that the signal peptide is present. How many amino acids? About 20 plus amino acids. The first 20 plus amino acids is the signal peptide. They clip between A and A-alanine and alanine here. So after, and then I will just remove the first 20 plus amino acids. This remove the signal peptide away from this sequence, and then use only the mature protein sequence to perform the subsequent blast. Okay, and then after perform the blast, and then I get the amino acids from various organisms. And this is amino acids from various organisms with different percentage of identity. Right? I choose 1, 2, 3, 4, 5 to get, including GDS acid is all together 7. Can I perform multiple sequence alignment using the first technique, cluster W2.1. This is example of what approach this one belongs to. Cluster W.
Progressive.
Progressive, yes. Yes, cluster W using progressive alignment. So you get this progressive alignment, cluster W, a progressive alignment to perform multiple sequence alignment. And then in order to check whether the alignment is correct or not, and then you have to download the published paper from the PubMed, and to double check whether the functional motif align at the same column or not. Okay, you can get this paper from the NCBI, and then you can double check. Okay, the column here, the block 1, block 2, block 3, and block 5. These are the conserved motifs belongs to GDSL asterisk. You see the dot on top indicate G glycine, G, and then D the molecule GDSL, right? And then the dot on top here indicate S and G H. This column from this block, the S should align properly, and then from this second column, the G should align properly, and then the other block, the N should align properly, and then the last block, the H should align properly. This is how you check whether the multiple sequence is correctly aligned or not, right? Okay, and then you have to mark in this multiple sequence alignment. You mark everything here. If they are conserved, and then you just mark so that I know you get the right answer. Huh? Okay, and then for the second practical 5.2, using cluster omega. The cluster omega is example of what? And what approach?
Iteration.
Cluster omega. No,
Iteration.
cluster omega. The cluster omega using HMM, hidden Markov model. So it belongs to which approach? Hidden Markov model, HMM.
Consistency based.
Yeah, the cluster omega belongs to consistency based approach, right? And then the same thing after you perform multiple sequence alignment, and then you also have to check and then mark the column that belongs to the functional motif, right? Same thing here. And then for practical 3 prime, and then okay, for this type of the multiple sequence alignment, you can check whether they align correctly by looking at the secondary structure. The red color is alpha helix, and then the blue color is beta conformation, and then they should align properly. Right? For example, this is helix, and then this part is beta, and then helix, beta, helix, beta. So this is how we judge the quality of the multiple sequence alignment using Praline, right? Okay. And then the last technique that you use is espresso. Okay, espresso is example of the, just now this one using secondary structure, but espresso using, this espresso belongs to which approach espresso used to perform multiple structure based. Okay, if there is structure used for the alignment, at the end, they will indicate which of the structure being used for multiple sequence alignment. Right. As you can see here, it gives you the signal here. If red color, meaning that it's good, and then yellow average, and then green color is bad. Okay, from here you see majority is good. Right. Okay, and then please take note. You see, from the template file list, if the program managed to associate with the original sequence, and then you have to check, you have to click on the template file list. If there is empty file, meaning that no crystal structure being used to perform multiple sequence. So after you click the template file list here, you see these are the output. So meaning that this structure, crystal structure, has been used to guide the multiple sequence alignment. Right? Only one. The same time, the GDSL also use this one, and then Vibrio also using the same of the crystal structure to guide the alignment. And the rest is no homologous crystal structure. So no structured information be used for the multiple sequence alignment. Right? You should have this as output in your report as well. So do you have any question for this practical?
Doctor, I have a question.
Yeah.
For the conserved sequence, the block, how do we identify that? Like, because the given is like the catalytic triad, right? So we just take 5 of it from there.
Okay. Okay, the block you can follow the paper here, and then you can just take the whole block here. So, okay, for example, the first block here, F, G, D, S, L, S, E, and then you just check your sequence, whether it consists of this or not. Right? If yes, and then you just highlight everyone, and then also use another highlighter to differentiate the S here. Because all of the GDSL accelerates, the first block, they must consist of GDSL. And then the serine, this is catalytic triad serine, should also conserve for all of the GDSL accelerates. And then you have the marks here, put a symbol on top, same as this, G, and the N, and the hash for this. And then you just follow how many of the amino acids in this block. Right? There are 4 blocks here. And then you just mark accordingly. Okay?
Thank you, Doctor.
Okay. Okay, and then next week will be your second test in the form of quiz. Since Dr. Fatiha will use a practical slot for her lecture, and then I will use the Tuesday slot for your quiz from 12 to 1. Okay or not? Next Tuesday, 12 to 1.
Okay, Doctor.
Only 2 chapter cover. Advanced database searching and also multiple sequence alignment. Just a very simple question. Right? Don't worry too much. The challenging question will be during the final exam. Right? Because it's unfair for you, because e-learning, so it's not easy to follow the lecture. Right? So first test, second test in the form of quiz, you should score. Right? And also the assignment. Okay, do you have any other question? If no question, see you on next Tuesday, 12 o'clock. And then for the next Tuesday, I will also post the attendance list to the WhatsApp group, and then you have to click to tick for me to record your attendance for the quiz. Right? Okay.
Okay,
Okay,
Doctor.
Doctor.
Okay, Doctor.
Okay, see you next week.
Okay, thank you, Doctor.
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 Thank you, Doctor.